Good afternoon. It's a great pleasure to welcome you in our summer time to a new seminar within the CRC 1182, Original Function of Metaorganisms. It's a pleasure to have Dr. Sebastian Hess with us today. Sebastian is an Emmy Noether Fellow at the University of Cologne. Before he gives his seminar and afterwards, he's happy to answer questions you might have. I briefly introduce Sebastian to you. He got his PhD at the University of Cologne. He studied there biology with a summa cum laude, then the PhD. Then he was a short time postdoc in Cologne, and then he moved to Canada, to Dalhousie University in Halifax. And with a number of different fellowships, he also was actually supported with a fellowship for his PhD study. And then he moved to Halifax with a number of different fellowships and was in all the major labs of the symbiosis kingdom, which is established there and around John Archibald and others. And after that, he returned back with a fellowship from the German Science Foundation before he then was awarded the prestigious Emmy Noether Fellowship, which he allows him now to have his own junior group at the University of Cologne. And in that position, I'm very happy to welcome you, Sebastian, here in Kiel in our collaborative research center. Sebastian made a spectacular observation in the last few years, and he published that very visible recently, and I assume that he will spend at least some part of his talk on that new discovery, which is and the lecture is entitled Physiology and Evolution of Multiple Endosymbiosis in Spirostomid Ciliates. Sebastian, the stage is yours. Please upload your talk, and after the talk, all participants here have the possibility, of course, to ask questions. We do it in a very simple way. Just raise your voice or raise your hand, and then you can directly interact with Sebastian. Having said that, we are now very excited and curious what Sebastian has to tell us.
Well, thank you very much for the invitation. It's a great pleasure to talk in front of this audience, and I'm surprised how well organized and visible this CRC is. Really great work. So, as Thomas already said, I'm currently based at the University of Cologne, and my field is the evolutionary ecology of eukaryotic microbes, and biotic interactions are really at the heart of evolutionary ecology of eukaryotic microbes. And I'm interested in biotic interactions, but why eukaryotic microbes? And there are three good reasons for this. These minute organisms are very diverse in terms of their lifestyles, their structures, and also their genomic material. They are ubiquitous. They really inhabit virtually every ecosystem on this planet, but they are still relatively poorly explored, especially when compared to visible macroorganisms. And today I'm going to talk about the physiology and evolution of multiple endosymbiosis in Spirostomid Ciliates. This is a project which was also mainly driven by a good friend and collaborator, Dr. Sergio Munoz Gomez. I have to mention him here. This was really work only possible with his effort on especially the genomic side of this study. Now, the title is quite specific, so I thought to give you a basic introduction. This is a simplified and schematic evolutionary tree of the eukaryotes, and I assume that some of you might not know every term because apart from the three big groups we see every day, the fungi, animals, and plants, most of the genetic diversity of eukaryotes is actually microscopic, if not unicellular. And we are talking about ciliates today, and ciliates belong to one of these branches. You see it here, the alveolates. And ciliates are interesting because they are very popular. Some of you might know the paramecium, but they are also, in my opinion, among the structurally most complex unicells. And they come in all sizes and structures, as shown here in this beautiful drawing, and most of them are actually heterotrophic. So they feed on bacteria or other eukaryotes. But not all microbes are heterotrophic, of course. So I want to talk a little bit about photosynthesis in the evolution of eukaryotes. And photosynthesis is widespread. That's the main point which I want to make today. And so I will just show where it occurs. And I think we all know about the plants, which are photosynthetic. And this goes back to a single endosymbiosis event about 1 to 2 billion years ago. But there are other related algal groups like the red algae and the glaucophytes. But that's not all. So here just showing very quickly this process. These primary plastids or chloroplasts go back on this endosymbiosis event involving a cyanobacterium. That's important because currently we imagine that it was a tiny and probably unicellular cyanobacterium getting involved and being basically the origin of the chloroplasts. However, this did not occur once in the evolution of eukaryotes. There is actually a single case of primary endosymbiosis with cyanobacteria, which is separate though. And this is Paulinella, an amoeba, which also took up a cyanobacterium and developed these chromatophores, seeing here these sausage-like structures. And they are not as far developed on the route of reduction to a plastid. They are kind of an intermediate organelle, so to say. Very interesting case. Paulinella sits here, by the way, so it's separate from plants and this lineage. There are also numerous secondary and tertiary endosymbiosis leading to most of the algae we know. That means a heterotrophic protist fed on a eukaryotic alga and implemented that as a so-called complex plastid. And this really gave rise to most of the algal diversity we see today. Taking this into account, already quite a fraction of the eukaryotic lineages contain members which are capable of photosynthesis. But that's not all. There is also the possibility of endosymbiotic or intracellular algae, and that's very widespread among protists, also among ciliates, as you can see here, that are all ciliates packed with these green algae, and thereby being able to utilize light indirectly. But these endosymbiotes also occur in amoebae and also in some animals, like this green hydra. I think you all know this organism very well. And these instances are marked here, for example. So in essence, photosynthesis is widespread, but interestingly, it all derived from cyanobacteria. And this is why all of the photosynthesis I showed you is oxygenic. It releases O2. And you might say, well, that's pretty normal. Plants do this all the time, and that is true. But from a prokaryotic perspective, from the cells which actually invented photosynthesis, this is quite an outlier. And this is nicely illustrated in this chart, showing the phototrophic prokaryotes grouped in oxygenic phototrophs, the cyanobacteria, and the anoxygenic phototrophs, those who do not produce oxygen, and many of them are actually sensitive to oxygen. They can't stand it. And there are different groups: purple bacteria, green bacteria, very rough terms, oftentimes comprised of different phylogenetic lineages. You see some of them here, purple sulfur bacteria and so on. And these bacteria are also widespread, but less visible most of the time, but you can find them, for example, in microbial mats in the marine ecosystems, where they live in layers below the oxygenic photosynthesizers. Again, they are very sensitive to oxygen oftentimes, but they also inhabit standing waters. Here, this is very impressive in my opinion. This is basically a column drawn from a deep lake. And when the oxygen tension is low enough and there is hydrogen sulfate, this badly smelling H2S available from decomposition on the ground, purple sulfur bacteria can flourish and produce these beautiful blooms. Now, this is basically the introduction to our study, because for some years now, we studied this beautiful ecosystem, the Simmelried, which is a peat bog near Konstanz in Germany. And in these standing waters, we also observe a curious pinkish sediment on top of brown plant debris and so on. And if you take a close look, you will see that indeed it is mostly composed of colonial purple sulfur bacteria. You can see these flocks here that are the actual bacteria, probably of the genus Lamprosystis. What, however, was most surprising to us, that there are also purple eukaryotes swimming around. This beautiful ciliate called Pseudoblepharisma tenue was described about 100 years ago, and it is pink due to interesting inclusions. And we had a very close look at these inclusions and found that indeed these inclusions seem to be bacteria. You can see the cells here, or also here, even in division. And interestingly, in the ciliate cell, they are combined with green photosymbionts, namely these green algae. And this is very strange, because green algae, as I mentioned, and purple bacteria have very contrasting physiologies. One produces oxygen, and the others can't stand it. And so we wondered, first, what is the identity of the symbiotic partners? And second, how do they live together? Unfortunately, we so far did not manage to cultivate this intriguing ciliate. So we did basically everything with hand-picked cells. And we developed two routes of sequencing strategy, involving freeze-thaw cycles of single picked cells, DNA extraction, and then a PCR-based approach for typical marker genes, and Sanger sequencing to really identify the partners. And in parallel, a whole genome amplification using the same DNA extract and Illumina sequencing to get genomic insights. I will first talk about the PCR-based approach and the actual identification of the partners. Now, this is a phylogeny of the bacterial sequence we got out of the Sanger sequencer. And interestingly, it falls in the group of the Chromaceaceae, that are the purple sulfur bacteria indeed. And it's a close relative of the well-known genus Thiodictyon. It's free-living Thiodictyon and occurs in standing waters. And these bacteria, as I already mentioned, they usually take reductive compounds like H2S, hydrogen sulfide, and use this as an electron donor for photosynthesis, thereby producing sulfur and other oxidized sulfur compounds. That's where the name comes from. A very interesting physiology, far apart from what plants or algae do. And we used the sequence and fluorescence in situ hybridization to target this 16S sequence. And we could show that indeed the many bacteria in the ciliate cell here shown in cyan belong to the sequence we got. So this was quite a good proof that we did not get the wrong bacterial sequence. We also did some single cell transmission electron microscopy and found that the bacterial cells in the cytoplasm are actually packed with these tiny vesicles. I hope you can see this. These tiny vesicles are usual for phototrophic bacteria. They are called ICMs, intracytoplasmatic membranes, or chromatophores, because that's where the photosynthetic apparatus is localized. It's basically increasing the surface for photosynthesis. And finally, we also checked for the typical pigments present in such sulfur bacteria or purple bacteria by hyperspectral microscopy. So we used an advanced microscope which can actually measure the spectrum of defined areas in the microscopic sample. And we were able to record the spectrum of algal cells and that of the pink bacteria. And we found that there is quite a strong difference. In essence, we found this chlorophyll peak for the algae and two typical bacteriochlorophyll peaks in the infrared region, which is typical for these purple bacteria. Another interesting aspect from this graph is you see that these spectra are kind of complementary. So we assume that there is quite little shading of the two very different photobionts happening. Now let's have a look at the whole genome data, because our main interest was also how does this system work and how do the algae and the bacteria live together. And this is a diagram. It's a circular diagram with the clustering here of the context we got. And you can see the samples here, P2, 4, and 5, which we sequenced. The coverage is shown here in black. And you can see some taxonomic clustering here in colors for the alga, chlorella, for the purple bacterium in purple, and for the ciliate in blue. And you see also that there is some difference in coverage between the samples. And this is because different extraction techniques. So we really tried to get every symbiotic partner, and it turned out that especially the algae are the hardest to lyse. So that's why we have different samples here. These metagenomes were actually quite complete, given that we used basically single cells. The most complete was the purple bacterium, actually. And it has some interesting genomic features. So this is the next or the closest known relative, which is free living. Candidate is Thiodictyon syntrophicum. And this is our bacterium in the ciliate. And as you can see, it has quite a genome reduction from about 7 or 8 in the free living relatives to around 3 megabase pairs in the symbiont, which is quite typical for symbionts, but it also shows that this symbiosis is really on a reductive path already. And this also applies to the number of genes. So from about 7,000 genes in the next relative to about 3,000 genes in our bacterium. Now, interestingly, there are a couple of typical pathways which are lost or defective in the symbiont. And this is, for example, the nitrogen usage. The symbiont cannot anymore fix any atmospheric nitrogen, hydrolyze urea, or store nitrogen as cyanophycine, which is very usual for many bacteria. But most stunningly, the symbiont also has a defective sulfur dissimilation. So there are genes for the oxidation of elemental sulfur, sulfide, and thiosulfate, which are either absent or pseudogenized and therefore not functional. And if we bring this in a context of the entire cell, we can conclude that this bacterium is really streamlined towards carbon metabolism. It has everything to do photosynthesis, it has everything to do the Calvin cycle and so on, and to use glycogen, and also to respire actually. But sulfur is not present anymore in this pathway. Interestingly, I mentioned the nitrogen usage, and we also found some transporters here, the glutamate aspartate transporter and an ammonium transporter, slightly suggesting that the bacterium might be able to import nitrogen compounds from the ciliate cell. And in the end, we were able to describe this interesting bacterium as a new candidate species, Thiodictyon intracellulare, the only known intracellular or endosymbiotic member of the Chromaceales, which performs photosynthesis, with a highly reduced genome and, interestingly, without sulfur dissimilation, which is otherwise a hallmark for these bacteria. So I want to conclude that through symbiosis, the ciliate, Pseudoblepharisma, really formed an unusual ecological niche by combining phagotrophy, that means feeding on others, like a ciliate, swimming around and so on, and unoxygenic photosynthesis. This is very rare. And we are currently trying to learn more about the detailed interactions. This, of course, is more challenging because we don't have cultures available, but there are different hypotheses. One of this is, for example, that under strict anoxia without oxygen and light, the alga is also able to ferment. Some algae can ferment and thereby produce, for example, atmospheric hydrogen. And this again could be used as an electron donor by the purple bacterium. However, there are alternatives, and it could also well be that this symbiosis is so fine-tuned that actually both the green algae and the purple bacteria can do photosynthesis at the same time. Now, this would work if the mitochondria of the ciliate host, for example, would be used for oxygen scavenging. This is a phenomenon known from many symbiosis, microbial symbiosis, that some byproducts are scavenged by other organelles or even symbionts to facilitate the performance of another compartment. Yeah? So in easy words, if the mitochondria respire enough, the cytoplasm of the ciliate could be anoxic enough for the purple bacterium to do anoxygenic photosynthesis, and still the alga could do oxic or oxygenic photosynthesis. Now, the next question we are very interested in: how did it come to this interesting tripartite symbiosis? And to tackle this question, we of course have to look at the relatives, ideally at the closest relatives. And we were very lucky to find an organism which is quite similar to the pink Pseudoblepharisma. You see the pink form here, but there's also a purely green variant in the same natural sample. And that's interesting, but not new. Actually, there was a variety described, again, decades ago, called Pseudoblepharisma tenue var pluraligera. And this is entirely green and packed with symbiotic green algae. And this species has a somewhat different lifestyle. While the pink form tends to dwell in the sediments, the green form forms these gelatinous tubes. It seems to be semi-sessile, so to say. And in these housings, it is basically creating a current to feed on floating bacteria. And you can also see this in the cytoplasm. There are food vacuoles with colorless content. So there's quite some evidence that this organism is as well phagotrophic. So it's actually a mixotroph doing photosynthesis indirectly with the algae and also phagocytosing food organisms. And by forming these interesting tubes, it can really grow to colonies, as you see here. It even looks a little bit animal-like, I have to say, when they crawl around in these colonies. Quite an interesting organism. A close look at these cells show that there are actually also some bacteria hidden between the algal cells. You can see this here already, but most closely here in this squashed ciliates. So that are also very big bacteria, colorless, between the algae. So this is also at least a tripartite symbiotic system. Very interesting. And the question arises, is it the same, just a different state of the same organism, or is it really different species? Now, we wanted to see if there are what all these tiny granules in the cytoplasm are, and we did a Lugol staining revealing these huge amounts of starch. All these tiny pink granules are starch granules, basically stained by iodine, suggesting that the ciliate really imports a lot of sugars from the algae. And now, where do these ciliates go? They belong to a genus called Spirostomum. This is a ciliate genus with, I would say, the largest ciliates known. And most of these ciliates are heterotrophs. So they are colorless, quite long, and they dwell in these muddy ponds in the sediment. Yeah? You can even see some ostracods, some real animals swimming around, that just illustrating the immense size of these organisms. And we were very lucky that we have an amateur microscopist called Thilo Bauer, and he is really a fan of Spirostomum. And together we are currently sampling all kinds of Spirostomum species to reveal the evolution of these ciliates, especially in terms of the endosymbiosis. Now, coming back to the phylogeny, you see that the two species, the purple one and the green one, are actually sister species. They are genetically distinct. They are not the same, but closely related. And there is another species called Spirostomum semivirescens, which also has endosymbiotic green algae. And we wondered how these organisms acquired the green algae. So we did a phylogeny and found that actually all these ciliates contain apparently the same genotype of chlorella. This is interesting because it slightly suggests that there is a single origin, at least in the Pseudoblepharisma lineage. And also very interesting is that it's the same genotype found in Hydra. So it could well be that these algae can quite easily escape a host, or just being released when a host dies, but then being incorporated in a totally different host. I mean, if you consider a metazoan like Hydra and a ciliate, that's quite a difference. Yeah, we also did some genomic sequences, and I will now cut a long story short. We got also the different partners, but we were quite surprised to find this, a bacterium not related to any sulfobacterium, but to the genera Accumulibacter and Propinio vibrio. They are closely related genera, And so we looked at these bacteria in more detail, and they are huge. Also, lots of storage material. We don't know yet what it is. We have some ideas, but this needs further testing. And there's one remarkable thing here, the genome coverage. We found that the genome has a coverage of about 200%, and this cannot really be. So struck by this observation, we did some further analysis and really found that there are two genomes which are very similar because these bacteria seem to be closely related, both stemming from the same ciliate cell. And well, we did a phylogeny with the 16S sequences and verified that there are two strains belonging to the genera Propinio-vibrio and Accumulibacter, and both of them, especially the Accumulibacter, show already some signs of genome reduction. You can see this here when you compare the genome size with of this symbiont to the next relatives. Yeah. But the question is, where is the Propinio-vibrio localized? Because we really didn't see this yet. And so we went back to the ciliates and did some DAPI stainings, and indeed could find highly DAPI positive structures, so with probably lots of DNA, but much smaller than these big Accumulibacter cells. So the morphological difference is really surprising. And we also verified this with some electron microscopy and found these very slender bacteria mainly localized to the cell cortex. So in the very outer region of the ciliate cell, there you can see these Propinio-vibrio cells, while the Accumulibacter cells are rather scattered around in the ciliate cell. So it seems to be that these two bacteria also occupy different locations in the same ciliate cell. Yeah, and that's basically a report about our ongoing research, and I want to end with some of the, let's say, conclusions we can draw for now. We know that there are at least two distinct species of Spirostomid Ciliates, which have very interesting endosymbiosis, oftentimes with more than two partners. Both harbor the same green alga, a chlorella, which was also found in the Cnidarian hydra, and probably goes back on a single symbiosis event in these ciliates. These ciliates tend to take up endosymbionts quite easily, so they have more than one endosymbiont, and it might well be that if we explore these ciliates further, we can even complete this picture of endosymbiosis further. And in general, we can say that through symbiosis, these ciliates really gain new abilities, especially the anoxygenic photosynthesis is remarkable, and by this form new ecological niches. Thank you very much. I'm open to questions.
Thank you very much, Sebastian. That's fantastic. It's a very clear and an amazing story. Just to remind the members of our CRC, we are mostly interested in interaction between microbes, bacteria, nowadays fungi with the host, which the microorganisms live outside. So Sebastian goes back to this classical symbiotic studies and endosymbiosis and understanding how that did evolve. Thank you very much. Questions are welcome, and just raise your hands or raise your voice. And as long as you get organized, I may just start, Sebastian, with numbers of questions, of course. But I would like to go back and try to better to understand the intracellular localization. You have the ciliates, then you have the algae, And then you have two types of bacteria. One of them sits in the outer, close to the membrane of the ciliate, and the other ones are packed, as I remember from the beginning of your talk, somehow. Are there anatomical structures, bacteriosomes, or something which really is the home for this bacteria, or are they just inside the cytosol of the ciliates?
Mm. Well, this actually can vary quite a bit between endosymbiotic bacteria. For example, the well-known Rickettsiales, they typically are freely in the cytoplasm, and but most of the endosymbionts have indeed a symbiosomal membrane. So they basically have a membrane around the bacterium which corresponds to, let's say, a food vacuole. Mm-hmm. Yeah. This is not yet so clear with the Propinio vibrio, because this was a very recent finding, and this needs further study. But it really looks like that it is stuck to the cortex, and you have to know that ciliates have a very special cortex. They are alveolates because they have alveoli, and that are vesicles which are basically fixed to the plasma membrane by the cytoskeleton. And sometimes it even looked like that the bacteria reside in these alveoli, which would be very interesting also from a cell biological aspect. But yes, you are right. Oftentimes they are in a separate compartment, not freely in the cytosol.
Mm-hmm. Very, very interesting. I really would like to thank Sebastian for taking the time to coming here and sharing his new findings on these fascinating symbiotic interactions between the ciliate, the algae, and the bacteria. Thank you so much, Sebastian, for coming, and best wishes and good luck for your future work.
Thank you.